about the next thing on my list, which is the difference between wide field and point confocals. And I want to say that these three lectures are quite heavy. There's lots of information and everything, but do not flip up. It's going to be okay because we're going to practice this a lot today. And you're going to have all sorts of quizzes and activities so that it all seeps into your brain. And for these two lectures that are coming now, this one and the next one, I actually have included my complicated animations on PowerPoint in Canvas. So if I forgot to share, I'm sorry. It's the usual thing for me. Somehow I always forget to share. There we go. So for these two lectures, if now you go to Canvas and you go to this, this lecture here, you will see not only the PDF, but you will also see the PowerPoint of the part that is animated because it's quite complex. I want you to be able to play it back and forth. So the PDF does not allow you to do this. Okay, so now let's get started with this. Click. Right. So once more, we're trying to talk about the light path. We've seen inverted, upright, and flat architectures. We've seen the difference between the microscope stand and the satellite equipment. And we have seen the difference between the light path in transmitted light and the light path in fluorescence. And now we're going to talk about the light path in wide field versus single point confocal. So as I want to remind you that we have what I call a system, which is the microscope plus all the stuff around. And this is the microscope stand. And this is all the equipment, satellite equipment. And luckily, we only need to identify the equipment that is connected through hard connection to the microscope body. And just if you can identify this type of equipment, then you can identify all types of microscope just by looking at them. Okay, so and this is quite simple. This type of equipment that we have, we need to be able to recognize a camera. And we will see that there's lots of different shapes. We need to be able to recognize a scanner. There's also lots of different shapes, and then a few more things. So two types of microscopes in the world. We don't care about those that cannot acquire images, but those that do not have a camera are the single point confocals that everybody calls confocals. Those that have a camera is all other microscopes. And if they do not have a scanner, they are a wide field microscope. If they have a scanner, they are a multi-point confocal or a spinning disk. And there's a few more things, right? All microscopes, all modern microscopes acquire digital images. So what is a digital image? So this is a digital image of the edge of a cell labeled with two different labels and imaged in fluorescence. An image, a digital image, is a matrix of picture elements. So if you wonder what pixel means it comes from picture elements. So it's a big matrix of a big collection of pixels. And each pixel has one XY coordinate and one intensity. So the intensity is a finite number of possible values. It means we cannot have just any type of intensity. For example, 50,093.5. No, we can't have that. So we have a definite number of possible intensities in an image. And that depends on the bit depth. So if we have an 8-bit depth, and we will talk about the bit depth later, but if we have an image that has been acquired in 8 bits, we have 256 values, and these are integer. So again, integer. So again, we cannot have like 20.8. This is just 0, 1, 2, 3, okay? So it depends on the bit depth. So we have a finite number of possible values. Now, if we zoom into this image, we're going to start seeing the pixels. And if we acquired the image with small pixels, we're going to see these small squares. And again, each of these squares has one XY coordinate and one intensity value. This is why across the square, we see the same intensity, okay? Because it has, if we acquire the image with large pixels, we see this even better. So it's like the same intensity across the whole pixel. So each pixel has a single X coordinate, single Y coordinate, and a uniform intensity. And we have this matrix. So throughout the image, we have tons of pixels, and each of them has these coordinates and intensity. So we need, on our microscope, a device that records the XY coordinates and a device that records the intensity. And that changes depending on the type of microscope. So in each image, we have a matrix of millions of pixels, but we also have something called metadata, and this is extra information. So you've heard about metadata before because I always ask you, you need to give us the images in the original format that the manufacturer of your microscope recommends because it preserves the metadata. And what can the metadata be? It's the information about the image. It can be, for example, what type of color you have assigned to this channel, what was the Z level in a Z stack, what were the acquisition settings, what did you have as like laser power and things like this, and acquisition time. And a lot of different information. It's very important that the metadata follows with the image, and this is why we want to be very careful with the format that we're saving the image in. So what is the difference when we acquire an image, a digital image with a microscope, with a camera or without a camera? There's a big difference. So if we acquire an image on a system that does not have a camera, it means this is a single point confocal, and there's only one type of system that acquires without a camera, single point confocals. In that case, the sensor, so we say the device that is recording the light, is called a detector. And it records only one pixel at a time, okay? One single pixel is recorded. And this detector always records images in black and white. And we will see why then we have our images in color. We'll see that later. On a single point confocal, we can have up to 34 detectors, right? Minimum 2 and maximum 34 per single point confocals. But each of these 34 detectors records one pixel, just that they record 34 different colors of the same pixel. So we will see this later, how we can image multiple colors at once using these different detectors. That will be for a different lecture. In a single point confocal, the detector only records the light intensity. And so we have only, the detector records only one pixel and only the intensity. And that means that we need to have something else that records the XY coordinate. And this is the job of the point scanner. So we'll see later how this works. But basically, you may recognize here some names of detectors. If you have a single point confocal, your detectors might be called a conventional PMT, it might be called a GASP PMT, HIDI, or APD detector. All these are types of names of detectors that we get on single point confocals. So now when we are talking about a microscope with a camera, is all the other systems except single point confocals work with cameras. So wide field systems, multi-point confocals that are also called spinning disk systems, or light sheets, and etc. all work with cameras. In that case, the sensor is called a camera, not a detector. And the cameras have several millions of pixels. So they do record several millions of pixels at once. And here is like a big matrix, boom, everything at the same time. We can have color cameras or black and white cameras, and we'll talk about that later. Why and what is the advantage of one versus the other. And we can have maximum 4 cameras per microscope. It's very rare, but it does happen. Most microscopes that work with a camera will have 1 camera. And so if you have 4 cameras, you get 4 images of millions of pixels. If you have 1 camera, you'll get 1 image of millions of pixels, all acquired at the same time. In a microscope that works with a camera, it's the camera that records both the light intensity and the XY coordinates. Unlike for single point confocals, where the detector records the light intensity and the scanner records the XY coordinates. Okay, so that's the difference as well. The camera records intensity and XY coordinates. Microscopes with cameras come with or without scanners. It depends what type of microscope we have, and then we will go through this. And these are the examples of the type of cameras that you may have on your system if you use a camera-based system. It can be called a CCD or an SCMOS, an EM-CCD, and they can be front illuminated or back illuminated. So just so that you can recognize the names when you hear about the EMCCD, maybe you didn't realize it was a camera. Okay. So most of you, sorry, most of you are going to be in a case where you have a single point confocal or a wide field, but I know that several of you this year although image with a multi-point confocal. And so now you know which one has a camera and not. So what's the difference when you sit by the microscope? Here is the difference. Now we're acquiring an image by 4096 by 4096 pixels on a single point confocal. This is what happens. And this is very, very nice. But as you can see, it is very slow. And this is why most people do not image at this with so many pixels. And now we move to a camera-based system. Pling! That's it. We acquire all these pixels at the same time. So that is a major difference between a single point detector system and a camera, is that the camera is much, 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 much faster. So let's see wide field systems. How do they work? So in this part of the lecture, I will compare wide field systems and single point confocals. So how do wide field systems work? Let's say we have a sample like this. It's some cells. They're labeled with several fluorophores, and we put this sample on the stage. And then we have the objective is here. And so can anyone tell me, is this an upright or flat or an inverted system?
Inverted?
Inverted.
Inverted because
The objective below.
Sorry.
Exactly. Very good. I like it. You guys are fighting towards her. It's brilliant. So the objectives are below the stage, below the sample. So this is an inverted system. Objectives are pointing upwards. Very good. So here we have this is the objective focal plane. So we are focusing the objective on this plane, which is where the sample is. Now there is no light on the sample, so there is no fluorescence and everything is black. But we come up with a light source, and that can be a mercury lamp or it can be an LED. In this case, and you see it's represented in white because mercury lamps and LEDs, they are quite broad in their range of wavelengths that they're emitting, unlike a laser, which has a single wavelength. So this is why I represented it in white. So we have some light coming out of the lamp, and it's white light. And now we use an excitation filter to filter this light to exactly what we want. And so now we get only the green light goes through the objective and to the sample. And if we have a wide field system that uses lasers, we do not need the excitation filter because this is monochromatic already. So we don't need to filter anything out. And in that case, we can skip the excitation filter. But for now, we're using a mercury lamp and an LED. So we have an excitation filter. And so this green light goes to our sample. And we have, as you know, a dichroic mirror. The light goes through the dichroic mirror. The sample is excited very well. This is wide field. It means the whole field of view is illuminated at once. That's why I mean like a big square of green light. The whole sample is illuminated. Oops, I go the wrong way. And this is why it's called wide field, right? Now, you can see in here in this really nice little picture that comes from Berkeley that you can see in here we have a little chamber full of a fluorescent solution. It fluoresces in green. And this is an upright microscope, right? Because the sample is above the stage. And you can see here, this solution allows us to visualize the excitation light. And you can see the shape of the excitation light is like a big cylinder that comes out of the objective. So it means that not only the plane of interest is illuminated, but everything above and below. So the whole plane, like here, the whole plane where we're focused is illuminated, but everything above and below is illuminated by this big cone of light, excitation light that comes from the illumination light source. So we keep this in mind. The shape of the illumination is like a big cone of light. And this illumination is defocused. It's very broad, all right? So we have a defocused illumination. All the X, Y, and Z in the sample are illuminated and excited. So here is a little question for you. What is the color of fluorophores that are excited by green light?
In the yellow, more or less.
In the yellow or in the red branch? Absolutely. So if you remember the fluorescence lecture that we had, we have the fluorophore and it gets some light in of a certain color and then shake, 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 shake. It loses a lot of energy through vibrations before it finally spits out a photon with the emitted light. And because of this loss of energy through vibration, the emitted light has less energy than the excitation light. And if you remember the graph showing the color of the light, compared to its wavelength and the energy, on the right side of the color towards the red and far red, we have less energy. And towards the left side, we have blue and green. There we have more energy. So a green light is going to lose energy, is going to excite fluorophores that emit in the orange or in the red, which is lower energy light. Okay, so very good. So now we have this green light that comes to our sample, and we're going to excite the red fluorophores inside our sample. And here we go. The fluorophores are excited, and they are spitting out some light, and the whole light comes back to the dichroic mirror. And as you know, the excitation light is eliminated by the dichroic mirror as well, but I haven't represented it here. So this dichroic mirror is going to reflect the emitted light towards the emission filter, which is in here in this filter cube. So we have seen all this before. The light goes through the emission filter, and because we have a defocused excitation, like this big cone of light, we also have a defocused emission. And we can project this on the camera with lots and lots of pixels. And then we get an image, pam, at once. So if the camera has a chip with 2048 by 2048 pixels, so that's millions of pixels, would be this multiplied by this. Then we get an image that is 2048 by 2048 pixels and everything up at once, okay? So this is what happens on the wide field system. And imaging with a camera is a little bit similar to flash photography. So you're in a room with your friends, you want to take a picture, it's a bit dark, so you put your flash. The whole room is illuminated with light. And then all that room will send back some light towards your camera, and you're getting the whole volume at the same time. So you're not only getting like the face of your friend that you focused on, you're also getting the back wall of the room. Okay, so it's the same thing in wide field microscopy. It's a little bit like flash. It's very fast, but you're getting the whole volume inside your image. So if you see here in this image, this is an image that would be acquired with a flash, with a wide field system. And oh yeah, I forgot this. So in this image is made of pixels. And if we zoom in a little bit, we can see that each pixel, as I said, is made of an XY coordinate and an intensity. So each of them. But that the intensity corresponds to the intensity that comes from all the Z, the whole volume at the same time. And this would be an image to compare. This would be an image acquired in wide field, and this would be an image acquired with a system that can do optical sectioning like a confocal. Okay, a wide field has no optical sectioning, so this is what you see. You see the whole volume. And if you have an optical section, the image may look like this. So I just wanted to show you the difference. So that was a wide field system. It's very simple how it works. Now we're going to look at how single point confocals work. So microscopes with detectors. So we have the same thing. We have our sample is on the stage. We have the objective. Now there's no light, so everything is black, and we have lasers. Currently, we have only one laser on, and it's the green laser. And then this laser goes through the scanner. And in the scanner, we find a dichroic mirror. Okay, so this dichroic mirror is going to allow the laser light to go through. This is why quite often on your single point confocals, you will see the dichroic mirror is named with the name of the laser because it transmits the laser. Okay, so the laser goes through, in this case, it's the green laser. It goes through the objective because I remind you that in fluorescence, both the excitation and the emission go through the objective, and it reaches the sample. Now we're only exciting a little spot of light instead of imaging, like exciting the whole field, we image a tiny spot of light. So it's a single spot. And if you look at with the same trick as the Berkeley University did, with this field with fluorescence so that you can see the shape of the excitation, you can see that in a single point confocal, the excitation light is focused on one point. And this is our focal plane. So this is where we are focusing in the sample, is at this little point here. But the excitation, the shape of the excitation is not a point, is a double cone, okay? So even though the highest intensity of the light is going to be at the focal plane, we clearly have quite a lot of light above and below the plane, okay? Which means that we are also bleaching above and below. Okay, so this is the a single point confocal. We have a focused illumination, and the shape of the illumination is a double inverted cone. So the excitation is a tiny spot of light, and also it's a double cone in Z. And this is the reminder of what it looks like on a wide field. We have instead a cone where all Z are excited. Right, so now our point of light is excited there and it starts sending red fluorescence as well. So the fluorescence goes back through the objective and it reaches the scanner again. And the dichroic mirror does its job by sending the light, reflecting the light, and it reflects the light towards the detectors. So now we have this red excitation light, emission light that goes towards the detector, and we can make sure that it has exactly the wavelength that we want with a filter. And the detectors in a single point confocal, we have lots of them. So we can have minimum two. In the simplest single point confocal, we have two detectors. And those that have most of them, most detectors have 34. Okay. So this emission light goes through, and we will see all this in this imaging multiple color at once lecture. So we go through, and now we slowly build the image point by point and line by line, because every time we're imaging only one point. So we have here a focused excitation, and therefore the emission is also focused. And that has enormous consequences of what we can do with a point detector. What we are going to do now is that we put a big metal plate right on the emission light path where the emission goes through, and we block most of the light. So big metal plate with a tiny hole in the middle, and this is the pinhole. So this is placed at one of the image planes on the microscope. If you remember the first lecture about optics, we have different image planes on the microscope, and the last one is the one that we are actually imaging. But this is placed on one of the image planes, and most of the light is going to be blocked. And then we have this the pinhole. So why do we do this? So you can see there's a lot of less light afterwards. Why do we do this? I will tell you in a second. But first, I want to tell you why is a confocal called a confocal? So we have the objective focal plane here, and we have the focal plane with a pinhole. And these two focal planes are conjugated. And this is the reason why a confocal is called a confocal, because we have two conjugated focal planes. The sample plane, which is the one where we focus in the sample, and the pinhole plane, where we have our pinhole and the big metal plate. That means that if you have a microscope without a pinhole, it cannot be called a confocal, because that's the definition of a confocal. You must have a pinhole conjugated to the object. So now, why do we do that? Why do we put this big metal plate there with a little hole? It's because it reduces the out of focus light. And this is why we have an optical sectioning. The single point confocal are able to do optical sectioning compared to wide field systems. And I will tell you afterwards how that happens. But for now, we have our sample little spot there that fluoresces. The emission light goes back through the scanner. And as I said before, the scanner is the one that records the XY information. So the scanner records the XY coordinates it records where we are there in the sample. And so it calls it X1 and Y1. And that light goes towards the detector, and the detector, or the one detector that is engaged, and I will show you later how we use several detectors. So currently we're only using one of all these detectors, and it records the intensity, which, if you remember, is also represented by lambda. In one point, just the intensity in this point, and then that's it. We get our intensity. Now we start building the image with it's a pixel with coordinates and intensity. Then the scanner scans the excitation light on the sample to a different spot. So here we go, and the scanner is going to move a little bit and send the excitation somewhere else. And so we have a distance between the first and the second spot, and this is the pixel size. The pixel size that you have in your image is the distance between the first spot to how far away the scanner has jumped, basically. So then that second spot is going to fluoresce, it goes back to the scanner, and we have the same thing. The scanner records the XY coordinates, and the emitted light goes through the scanner and through the pinhole, bloom, we go again, and we record the intensity. Here we go, and we add another pixel. And now we do that again and again. We move again, and then we get another pixel, and then we keep going like this. And we build the image slowly, point by point and line by line. Okay, so this is the way a single point confocal works. This is when we are only imaging, when we're doing sequential imaging, which means we have only one laser on, and we also have only one of all these detectors is actually collecting the information. So this is sequential imaging. But as we know, on a single point confocal, we can actually image with several detectors because we have so many of them. So we can also image simultaneously. When we do simultaneous imaging, we're going to have several lasers at the same time and several detectors at the same time. So how does that work? So now we're adding a new laser to our system. So now both the green laser and the red laser are on. So what is the color of fluorophores that are excited by red light? Anyone?
Infrared, I guess.
Yeah,
Far
before
red.
the infrared, far red. So between the red, we still have quite a bit of space to the before we go to infrared. So infrared and UV are outside the visible range. A red laser is about 640 nanometers or something like this, and so we have the far red. It's still in the inside the visible range. So it would be a far red laser. A far red, sorry, fluorophore that is excited by red light. Good. Now we have basically we have the two lasers on at the same time, which means that the two fluorophores are super well excited because we have the two lasers on. And so they both fluoresce, and what we get back from the sample is a complex light with the fluorescence from two different fluorophores. Okay, and that goes through the objective, reaches the scanner, and then the scanner is sending this complex light towards the pinhole and towards the detectors. And now we have two detectors that are on at the same time. And what we have is here a dichroic mirror. Because if you remember, we have 4 roles of the dichroic mirror, and the 4th role of the dichroic mirror is to split the emitted light, complex emitted light, into its different components. So now we're going to have this dichroic mirror that is going to send the far red light towards a detector and an emission filter and one detector, and then the red light towards another one. So we can separate them with an extra dichroic mirror there and add some emission filters. And each of the detectors is going to, at the same time, simultaneously detect the two emission. And you understand that this is the case where you really want to be very careful because the 2 fluorophores are excited super well. So you want to be very careful that you have no bleed through. Otherwise, you will not be able to see the correct fluorophores in the correct image. So this is what happens when we are imaging simultaneously. But we have spoken before about fixed and virtual emission filters. And this is on the single point confocal. So what is that? If we have a filter like this, it's a fixed filter, but we can also have so what happened there? Yes, I went the wrong way. We can also have something that I will represent this way. Different companies have different ways to use virtual emission filters. They have different designs, let's say. But basically, you can think about this as a filter that would work like a rainbow and that can move in this direction. And so if it's in here, it's going to only allow green photons to go through. If it's in here, it's going to allow red photons to go through and everything like this. So means using this, you can have the same filter, just move that thing, and this is a virtual filter. But again, we will see later in another lecture how that actually works. But in a single point confocal, we can use fixed or virtual emission filters, and that's it. So that was basically how a single point confocal works. And I hope that slowly things are falling into place for how your microscope work. But don't worry, you will become an expert. Now, how do pinholes reject the light? I'm just checking how I'm doing time-wise. I'm okay. How do pinholes reject the autofocal light? So we have seen that in a single point confocal, the excitation is a single point. It's a focused excitation, and it's the scanner that sends the laser there, and then the scanner also sends the emission towards the pinhole and towards the detectors. So we have this shape of excitation on a single point confocal. Oh, my fish has moved. It should be in the middle. Anyway, never mind. And so if you remember our optics lecture, this is an iris that we have placed at an image plane. So the pinhole is actually an aperture in the light path. And on a single point confocal, you can open it and close it. It's something mechanical that you can open and close. So you can change the size of the pinhole. And what happens there is that we have an image. So what does the image of a tiny point light source look like in the microscope? You may remember from our optics lecture that this is what it looks like. So we have a tiny, tiny light source, which is exactly what we have here because we excite just a little point. And this sample, this little spot in the sample acts like a light source because it fluoresces, and it fluoresces in all directions. But some of this light will make it to the objective. And through the objective, because of diffraction and interference, we have the microscope forms an image of this tiny spot of light. And the image does not look at all the same as the tiny spot of light. It is much wider, so it's enlarged in XY and it's elongated in Z, so it looks like a rugby man. And this is the rugby ball. And so we have a bright center, elongated in Z, and we have all sorts of rings around. This is the point spread function. The point spread function is the image that the microscope forms of a tiny point source of light, and like we have on the single point confocal. And if we would draw a line in here and we can see the profile, we see here the blob in the middle, the bright, very bright center of the point spread function. And here we see sort of the waves that are formed by these circles. Okay. So now what we do when we put the pinhole here, since we are in the image plane, we block all the light that is outside. So I show you all these things here is going to be to disappear because we have just this in the center. And if you see it here on the in the Z dimension, it's the same type of thing. So we're going to block all this. You can imagine the plate comes across perpendicular. And so we're blocking, basically what we're doing is that we're removing all the rings. We're keeping only the bright spot in the center. So this is what it looks like once we have our pinhole. But now if we have a fluorophore, this is, let's say, one fluorophore, but if we have another fluorophore that is somewhere else, it's in a different Z plane, what will happen? It's like it's this. And the light that comes from the bright center of the fluorophore image, the point spread function, is going to be blocked by the pinhole. And only a little bit of light coming from the ring is going to participate in the image. So this is the way the pinhole works. It actually eliminates the bright centers of the other point spread functions that are out of focus, and it only keeps the one that is in focus on the focal plane, right? So out of focus and in focus. So the out of focus light is greatly reduced. It's not completely eliminated because we get all the rings, but this is very little light compared to the center. The center is much, much brighter. So only the light from the in-focus fluorophore is sent to the detector thanks to the pinhole that, well, the big metal plate that throws away everything else. Okay? So I just want to point out that in a single point confocal, we do illuminate a double cone, but we only send the stuff from the focal plane to the detector. This means that we are bleaching above and below the focal plane, but we throw away all the light that come from above and below, okay? So now I want to tell you the similarities. Yes, I want to tell you the similarities between a wide field and a single point confocal. So both of them can be on an upright or inverted microscope. You can have transmitted light imaging on both of them. You can have fluorescence imaging on both of them. And they can both be fully automated, very advanced systems. And you can do Z-stack on both of them. And also you have a high optical resolution in XY on both of them. Okay, this is something important to remember. In a wide field system, we anyway have a high optical resolution in XY. We can do fixed and live, image fixed and live samples on both of them. And in a wide field system, the whole volume is bleached, whereas in the single point confocal, the whole volume is bleached, but it's not like it's like a double inverted cone. So the shape of the illumination is different between a wide field system and a single point confocal. And we can do super resolution in both wide field system and single point confocal. It's different techniques, but both of them have that. Now, have a look at the differences between wide field and single point confocal. A wide field system uses a camera with millions of pixels, whereas a single point confocal has a few detectors, all of them image the same pixel. Okay, so we image only one pixel at a time. So wide field system is a little bit like a snapshot, like a flash photography, whereas on a single point confocal, we scan the sample point by point and line by line. So it's much slower. Wide field has a wide field illumination, so it's a cylinder illumination, whereas the single point confocal has a focused excitation light with a double inverted cone. That's the shape of the illumination. A wide field system clearly is very fast compared to a single point confocal, which usually is slow. And then in a wide field system, because in general we have only one camera, usually we only acquire sequentially one color and then another color and then another color. But in single point confocals, because we have a few detectors, we usually can combine multicolor channels and image simultaneously. A wide field system has no pinhole. It is very photon efficient because we're not throwing away any of the fluorescence that comes from the sample. Whereas a single point confocal, we have this big metal plate with just one hole in the middle. So we throw away all the light that comes from the outer focal plane. But the disadvantage of not having a pinhole in a wide field system is that we are recording all the fluorescence that comes from all the Z levels at the same time. And so the image may be a bit blurry if your sample is just a tiny bit thick. Whereas in the single point confocal, we have an optical sectioning capability, and only one Z level is recording. The fluorescence that comes from the autofocal plane is eliminated by the pinhole. A wide field system is a low contrast in Z. This is because we have this blur here. Only in Z, right? It's high contrast in XY, but low contrast in Z for a sample that has more than a very, very thin layer. Whereas it has a high contrast in Z, even for thick samples, thick fluorescent samples in the single point confocal. So the resolution in Z is not so usable, let's say. We have good resolution, but we cannot use it without deconvolution on a wide field system. Whereas on a single point confocal, we can use the high resolution without needing deconvolution. And we will talk about deconvolution later, and you will try as well. On a wide field system, we have a low flexibility, and I will explain later why. And on a single point confocal, we have a very high flexibility of changing the pinhole and the pixel size. So we'll talk about that later, but high flexibility is one of the major points of single point confocals. On wide field system, we're super sensitive, so we can image single fluorophores, whereas we are in most cases lower sensitivity in a single point confocal. Again, we will talk about that. And in wide field, we have no spectral imaging, so we have to image sequentially only, whereas in single point confocal, we can use these very nice filters that slide and that we can call virtual filters or spectral imaging with different techniques. A wide field system is simpler and it's also cheaper. And of course, for a single point confocal, it's the opposite. They are anyway expensive. Huh? But so the main differences between a wide field system and a single point confocal is a wide field system is super fast. A single point confocal is very flexible. So these are the advantages really of both of them. And there are other ways to remove the autofocus light during acquisition. And so removing the autofocus light is called optical sectioning. So we can use a multiphoton, but we are not going to talk about this in this course. A multiphoton also have a optical sectioning capacity. We could use a multi-point confocal with multiple pinholes, and we will talk about this in the next lecture. This is also camera-based. And we could use a light sheet system, which is also camera-based. So that's it. So for this little lecture, the conclusions are that wide field systems use a camera with millions of pixels, and they image the whole field of view at once. They are very fast, but they do not have optical sectioning capacity. Whereas single point confocals use detectors that image only one pixel at a time, so they are slow. However, they have an optical sectioning capacity and they have several detectors to help. So we can do simultaneous acquisition. And they also especially have a great flexibility that allows spectral detection and an adjustable pinhole and pixel size. So all these details here, I will explain to you later. Okay? Right now we're focusing on how does the machine works. So I put the PowerPoint of these animations that I see are quite complex. So they are in Canvas and you can look at it. So now I'm going to stop